This is part 2 of the cell. Today we're going to talk about the nucleus and the ribosomes. The function of the nucleus is to contain the eukaryotic cell's genetic library. That's where the genes are located inside the nucleus. Some of those genes can also be located in the mitochondria and the chloroplast, but we will talk about that when we talk about those particular organelles. The structure of the nucleus, it is surrounded by a double membrane, just like the regular cell membrane. It is sometimes called the nuclear envelope. It's also called the nuclear membrane. It is a double membrane that is fused in spots to form pores, which allow certain molecules to pass through, some larger macromolecules, and some other particles to pass through that membrane. As you see here, this is the fusion between two of the two double layers there, and you can see this pore, and there's a whole bunch of those pores on the nucleus. This is a process. This is protein synthesis right here, but you can see that these pores are used to take messenger RNA out of the nucleus. Remember, DNA cannot leave the nucleus, so it has to be transcribed into a piece of RNA, and then the RNA leaves the nucleus. So that's one of the items that leaves through those nuclear pores. The DNA itself is organized into a fibrous material called chromatin. It normally appears as just this kind of mass inside the nucleus. We'll talk about the nucleolus next. The only time that chromatin material becomes organized in these particular structures here called chromosomes is when the cell is preparing to divide and undergo mitosis. But for the most part, it is just this kind of purpley mass here called chromatin. That dense spot in the middle is called the nucleolus. The nucleolus is responsible for producing the ribosomes. It makes rRNA, and that rRNA combines in different configurations here to actually create the ribosomes, and those must also pass out of the nucleus through those pores. So it has to be big enough for the ribosomes to be able to pass through. The ribosomes are responsible for making proteins. The structure of them is that it is made of the rRNA that we just talked about that was produced inside the nucleus. There are two subunits. There's the large subunit and the small subunit. And when this reads the genetic material to actually make the protein, the genetic material is kind of fed through the middle of those two subunits. There are two different types of ribosomes. There are free ribosomes that are just inside the cytosol, and they synthesize proteins that specifically function within the cytosol. Then there are other ribosomes that are considered to be bound ribosomes, and they are attached to the outside of the endoplasmic reticulum. The endoplasmic reticulum is responsible for making proteins that are exported out of the cell or that are going to become part of the cell membrane, as pictured here in this little cube. These are proteins that are integrated into the cell membrane, and those would be made on bound ribosomes. Again, we're going to talk about the endoplasmic reticulum in a different presentation, so we will come back to that. Prokaryotes and eukaryotes do have different ribosomes. Structurally, they are different from each other. They still have a small and a large subunit, but the sizes of those are different. They are made of different proteins. Prokaryotes and eukaryotes have very different functions. Prokaryotes are very simple. Eukaryotes are much more complex and do a lot of different things, whereas the ribosomes in the prokaryotes are not quite as complicated. The other thing to keep in mind is that there are no free versus bound ribosomes in a prokaryote. Prokaryotes do not have endoplasmic reticulum. so they only have free ribosomes floating around in them. And I'm going to leave you with this last final thought, and that is it.